Okay, well, welcome back, and let's begin. So my name is Bob Dorsam. I'm director for the Division of Pharmacology Toxicology Review in the FDA's Office of Generic Drugs, and I'm moderating the session. The session's title is Safety and Risk Assessment of NDSRIs for Human Health. By way of introduction, studies published decades ago tell us that nitrosamines such as NDMA, NDEA, pose mutagenic and carcinogenic risk. The risk is compound specific, and several factors may contribute to either increased or decreased potency of a nitrosamine. Much of the published literature is on nitrosamines that are relatively small in molecular weight, as you heard from Dr. Raw earlier this morning. And more recently, we know that NDSRIs are at risk of forming in some drug products, and so recently, research and collaboration has been directed towards properly characterizing the risk of these NDSRIs. Since NDSRIs are data poor, we're very much looking at what is the appropriate way to assess the risk. And so today's session will cover several evolving areas of risk assessment for NDSRIs, and I would put it into three buckets. One of them is on optimization of the Ames assay, the other being on structure-activity relationships, and then the third is on gaining a mechanistic understanding of NDSRI's mutagenic potential. So for this session, the first talk will be given by Dr. Robert Heflich from FDA's National Center for Toxicologic Research. He will discuss his group's research on optimizing mutagenicity testing for nitrosamines. The Ames assay is one of the key assays that we use to characterize a compound's mutagenic potential, and literature has suggested that the conditions of the Ames assay may result in different levels of sensitivity for when we're assessing a nitrosamine's mutagenicity. His work is looking towards optimizing those conditions. Considering that NDSRIs often do not have data to characterize their carcinogenic potency, the chemical environment surrounding the nitroso group is critical in order for us to estimate the potency of that compound. So Dr. Ponting from LASA will discuss the mechanistic rationales for the effects of structural features on the activity of nitrosamines. And similarly, Dr. Naomi Kruhlak from FDA will discuss structure-activity relationships to inform setting acceptable intakes for nitrosamine impurities. Lastly, Dr. Raphael Nudelman from Teva and Dr. Andrew Teasdale from AstraZeneca will discuss their mechanistic studies for NDSRI-related mutagenicity. Now, following the presentations, we will have a 30-minute panel discussion with our speakers. We will also be joined by Dr. Sruthi King, who is Deputy Director of the Division of Pharmacology Toxicology Review at the FDA. Although I had originally planned to go through questions and then open it up to the audience, the discussion coming from the audience in session one was really quite good. And so what I would encourage you all to do as we're discussing questions, if you should want to interject, have a clarifying question or comment, I'd encourage you to come to the microphones at your will while the topic is being discussed. Okay, so on to the presentations. We'll first begin with Dr. Robert Heflich from NCTR.
Okay, well, thank you for the introduction, Bob. I hope you can hear me.
Yes, I can hear you. Thank you.
Good, that's a good start. And I'll try sharing my screen. And let's see if I can minimize this window. There we go, everything's going miraculously well. Okay, as Dr. Dorsam said, my name is Bob Heflich, and I am a FDA employee, but I am at the National Center for Toxicological Research. So I suspect a lot of you know what that is, but it's a part of FDA that has no regulatory duties, but our role being to provide research studies that might be useful for FDA in making their research decisions. And the issues surrounding nitrosamines and the fact that genetic toxicology is a key endpoint for my nitrosamine evaluation sort of provided us with that opportunity to do some studies in the last few years. I direct you to the fine point at the fine print at the bottom of the slide. I think you have heard this disclaimer before, but you should sort of double it for me because I'm not a regulator and I often say things that I probably shouldn't. So just be aware that whatever I say do not represent the views or policies of the US FDA. I also should note that what we have been doing with nitrosamine mutagenicity is also is only part of what's going on in the field. There are several very large groups, multi-laboratory studies going on. I'll just note the HESI project that involves a lot of pharmaceutical and CRO people doing something very similar to what we're doing, and the EMA studies going on in Europe. So hopefully what we're doing will contribute to addressing the safety issues associated with nitrosamine drug impurities. So I've divided our nitrosamine work into 3 categories. We started about 3 years ago investigating the mutagenicity of different nitrosamines in the Ames test. The object of the Ames studies is to examine ways of enhancing the detection of nitrosamine impurities with the objective of optimizing the assay for the detection. We also have made a considerable effort with in vitro mammalian cell assays. In our mammalian cell studies, we have concentrated on using human cells that have endogenous human metabolic activity. And I'll cover these first 2 points in this talk, but we also have started some in vivo work using the big blue rat model. The objective here is to evaluate different in vivo genotoxicity endpoints, including the transgenic mutation endpoint, error-corrected NGS, and PIGA mutation, in order to determine which are the most appropriate for detecting the genotoxicity of nitrosamines in vivo. So Bob already covered a lot of this in his opening remarks, but I just want to indicate that nitrosamines as a class of test compounds has the reputation of being somewhat finicky or difficult to detect in the Ames test. And that's somewhat because of the various ways that the testing has been conducted over the years, and sort of people publishing all these very interesting and sometimes conflicting findings. And it's possible to get negative findings with potent mutagenic nitrosamines that are fully using a test that's fully compliant with the OECD test guidelines. And as Bob also said, another issue is that very little is known about how these problems with the Ames test relate to NDSRIs, which are recently recognized class nitrosamine impurities from drug substance itself. I'm sure you all know that. That's why we're here. So thus there's a need for a version of the Ames test optimized or at least enhanced for detecting nitrosamines that will increase the regulatory agency's confidence in test findings. So as Bob also said, people have been studying nitrosamines for quite a long time, and going back 50 years to the beginning of bacterial mutagenicity testing. I like to refer to a paper by Heinrich Balling that was published in 1971 that, as far as I know, was the first paper that indicated the use of S9 for activating a pro-mutagen to something that was mutagenic in bacteria. And it actually used the nitrosamine NDMA to demonstrate that. And here's a list on this slide of a number of the protocol choices, mostly compliant with test guideline 471, that one can apply with testing of nitrosamines, and that seem to have different effects on the mutational yields in the assay. So what we've done in our project is to test several of the most promising of these protocol choices on a series of nitrosamines, including NDSRIs, about half NDSRIs, half small nitrosamines. And a general description of what we've done is described in the sub-bullets of the slide. We used 5 tester strains, 5 metabolic activation conditions, 2 pre-incubation times. We didn't use the plate incorporation assay. And we've made attention to solvent choice and concentration of solvent. The initial goal was to test 28 nitrosamines and NDSRIs. And we're just about done. Actually, this week, we're just about done with running through those 28. So we'll have some, I'll present some initial, some preliminary findings here, but we will have a full set of data to show you in hopefully in a couple of months. So here are some big picture results from 13 of the NDSRIs that we've screened in our Ames testing. I've coded them for this presentation. 7 were positive in our Ames screen. 1 was direct acting NDSR8, but S9 increased its activity, and the direct acting activity and the S9 mediated activity were in different tester strains. We've used a hybrid of the fold rule and the BMD dose response ranking to compare the extent of the responses. And I think you can see a pattern in the positive responses in terms of the most sensitive tester strains in the middle column and the most effective activation systems in the rightmost column. I'd like to add that while most of the nitrosamines we tested follow a very similar pattern, there are about, I'd say 15% of them that give unusual responses. So there are some surprises. And as Bob said, there's very little cancer data associated with the NDSR cancer tumorigenicity of the compounds that we've looked at so far. So to move on to our in vitro mammalian cell studies, as indicated on this slide, the motivation to conduct these studies came from some comments made at a public workshop held by CEDAR about 2 years ago. The comments indicate that data from human cell systems using human metabolic activation paths would be useful for following up on Ames test data. We happen to be working with 2 such assays at NCTR that seem like they might have relevance. One is a human lymphoblastoid TK6 cell system consisting of a series of 14 cell lines, each transduced with a different human CYP. And I'll show you what those are in a minute. And then there's the human HEPA-RG human cell, liver cell line that a lot of people are excited about, which expresses a range of human phase 1 and phase 2 enzymes. So this slide compares the 2 systems we're using. I like to think that they're complementary in some ways. The TK6 cell system utilizes 1 human CYP at a time for activation, which is strictly phase 1 activation with no cooperation between CYPs. We've used these cells to do DNA damage studies with COMET and the multi-flow assay, micronucleus analysis, and we've also evaluated the HBRT and TK mutational endpoints in these cells. On the right, the HEPA-RG is a human hepatic progenitor line that can differentiate into mature liver-like cells. And the differentiated cells then can be induced to divide which is a useful property for evaluating some of the genotoxicity endpoints like micronucleus and gene mutation. As I indicated previously, these cells express a range of phase 1 and phase 2 enzymes, not unlike the human liver, but several of the CYPs are expressed at relatively low levels. And as it turns out, I've listed them here, this could impact HepRIG cells' sensitivity to some nitrosamines. We've used the HepRIG system to evaluate DNA damage with COMET and multiflow, perform the micronucleus assay, and we have some promising results performing mutation studies with these cells using error-corrected next-generation sequencing. So here's our initial thinking on the value of these assays. Most of what we know about human risks associated with nitrosamines is based on rodent tumor bioassays and mutation assays conducted in bacteria with rodent liver activation. These human mammalian cell assays could be used to confirm or study these findings, both positive and negative, in a human-based system. And they can also be used to assay compounds like NDSRIs based on antibacterial compounds that are now readily tested in the Ames test. The mouse lymphoma assay could also be used here, of course. And the TK6 system gives mechanistic data that may be useful to understanding the mutagenicity of the compounds. I'll show you some of that in a minute. Well, by combining phase 1 and phase 2 activities, the HepRIG system takes a step beyond hazard ID that may be useful for contextualizing human risks. So I'd like to spend some time describing these two in vitro systems. The TK6 cell system studies have been conducted by Shilin Li, Wan Lei, and they've been conducted under the direction of Nan Mei, the PI on the study. As I said, there are 14 of these lines, each expressing a different human CYP. And here are Western blots to demonstrate that. And you can see the CYPs listed on this slide. And at the bottom of the slide are a few references that give some information on development of these lines. Before we started using the system for nitrosamines, a number of different toxicants have been evaluated with these cells, and they're listed on the right, a variety of different toxicants. And a number of endpoints have been evaluated, listed on the left. I've highlighted the endpoints relevant to the nitrosamine studies. And there are a couple of references listed below that provide background information on the studies on the compounds listed here. So Shilin and colleagues published a paper last year that used the CYP2A6 expressing line to evaluate the genotoxicity of small to medium-sized nitrosamine drug impurities. These were listed in the recent FDA guidance to industry. Unfortunately, I neglected to add that reference to the slide, but the paper is in last year's Archives of Toxicology, and Lee is the first author, Li is the first author. What I wanted to show you in this presentation are some data from an NDSRI. It's N-nitrosopropanolol, which was listed as NDSRI 8 in the table with Ames results that I showed previously. It was the one that showed both direct acting and S9 mediated activity in the Ames test. This figure on the slide was taken from a paper that just was published in Regulatory Toxicology and Pharmacology, and the references at the bottom left. Doing mutation assays with 14 strains of cells is a little impractical. So what Shilin did in the N-nitrosopropanolol study is to screen for genotoxicity in the CYP expressing lines that most likely activate nitrosamines. And it was the screening involved using the relatively high throughput flow cytometry-based micronucleus assay. Those responses for 9 cell lines are shown on the slide. 8 lines transduced with a single CYP, along with the parental non-transduced TK6 cell line. You can see that N-nitrosopropanolol produced quite a strong response in one of these strains, circled here in red. This is a strain expressing human CYP2C19. I don't know if you can read the concentrations on the slide, but the concentrations producing a positive micronucleus response in the CYP2C19 expressing line were all less than 1 micromolar, while most of the other assays shown on this figure were conducted with much higher concentrations. Following this screen for genotoxicity, HPRT and TK mutation assays were conducted, and they confirmed that N-nitrosopropanolol induced mutations as well as micronuclei in this CYP2C19 expressing line. All this data is in the paper. One further point that can be made from these data, you will recall that N-nitrosopropanolol was a direct-acting mutagen in the Ames test. Note that the upper left-hand panel in this figure, that N-nitrosopropanolol was negative in the non-transduced parental TK6 cell line. So while N-nitrosopropanolol was a direct-acting mutagen in Ames, it was not a direct-acting genotoxicant in human TK6 cells. So Sheelan, wrap the last slide up there, Sheelan and colleagues are gathering data on the other 15 or 16 NDSRIs that we've tested in Ames. So eventually there'll be a lot more data to report. To move on, this slide shows, introduces the work we've done in HepRG cells. What I will describe is mainly the work of Jian So and Xiaoqing Gao, and Hannah Xu, also listed here, has joined us recently as a postdoc. I described HepRG cells as a human hepatic progenitor cell that can be differentiated into mature cells that behave like human liver cells. An interesting observation about these cells is that the mature cells can be cultured in 2 ways, as attached cells, shown in the left of the slide, and as spheroid aggregates of cells, which are shown on the right in this. And this is a photo of the wells of part of a 384 well ultra low attachment dish. If you look carefully at the individual wells, you can make out little pinpoint cell aggregates of several thousand cells. These are what are called spheroids. The significance of spheroids is that they tend to be functionally more liver-like, including expressing higher levels of phase 1 enzymes than do the attached cells. In this bar chart, the filled bars represent mRNA expression for spheroids, the open bars for attached cells. Note that there are higher expression levels for the phase 1 enzymes in spheroids than attached cells. The y-axis is on a log scale, so the differences are in the 5 to 10-fold range. Note also that there are a few CYPs that have relatively low expression, including CYP2A6, which is involved in the activation of low to medium-sized nitrosamines. And if you compare those bars to the bars with CYP2C19, for instance, you can see a rather large difference. And you recall that CYP2C19 was important for the genotoxicity of N-nitrosopropanolol. Note also that phase 2 expression is similar or even a little lower in spheroids compared with attached cells. So this is how Jian and Kruhl are setting up experiments. For the studies to date, they have been treating both 2D and spheroid cultures and comparing the responses. As I indicated, the spheroids are made in 384 well low attachment plates, but the treatments for both the spheroids and 2D cultures are conducted in 96 well dishes, which are shown at the top middle here. Treated cells are used for the common chip assay, as well as given EGF growth stimulus, and then at which point they start synthesizing DNA and dividing. And then the assays for the micronucleus and further DNA damage endpoints are conducted. And as you might guess, mutation analysis would be done after growth stimulation. So here's an example from one of Gion's papers of the remarkable differences in sensitivity that could be seen for nitrosamines tested in assays using spheroids as opposed to attached cells. The example here uses common analysis of NDMA, NDMA-treated 2D and 3D cells. The increasing red line represents responses in spheroids. The more slowly increasing dashed black line in attached cultures. Presumably, the DNA damage response is mediated by CYP2E1 activity, which you may have noticed is a bit higher, but not really substantially higher in spheroids than in attached cells. And here are the common responses produced by our example NDSRI and nitrosopropranolol. Again, different, but a lot more subtle a difference than seen with NDMA. So Hannah, our postdoc, is busy putting our set of NDSRIs through this assay. So eventually we will have a nice data set to look at. And as you can probably guess from the last couple of slides, these data lend themselves to BMD ranking analysis. Here are some results from a screen of 8 low to medium-sized nitrosamine impurities in the HEPO-RG cell assays for the COMET endpoint, micronucleus, and a little data from spheroid data for the gamma H2AX endpoint in the multi-flow system. The horizontal lines are confidence intervals for BMDs estimated from dose response data, with the more robust responses trending to the left side of the plots. I think you can see that the COMET and micronucleus assays for the spheroids, shown by the confidence intervals in red, are generally more robust than for the 2D cultures, whose confidence intervals are shown here in black. And you can see that the response for NDMA were the most robust, at least for the COMET and micronucleus endpoints. NDMA responses are about halfway down in the figures. So finally, here are some of the people who have been involved with the projects. On the left, the people from NCTR, I already mentioned those involved with the in vitro mammalian cell studies. So the Ames test people, Bo Middlestat was the lead until she retired last year, at which point Michelle Bishop took over. Tao Chen, Jian Yan, and Javier Ravallo are involved in the in vivo studies. On the right are some of our collaborators from CEDAR. Asa Ostracci and Tim McGovern have been the leads, but all those listed have made important contributions. Of note is Rosie Ellisbrew, not from CEDAR, but an FDA expert in nitrosamine genotoxicity and the Ames test. Unfortunately for the project, Rosie retired from FDA at the end of last year. And thank you for listening. That's all I have. Although this slide asks for questions, I guess we're going to field questions at the end of the presentations. So I'll turn this back to Bob. Stop share. There we go. And I'll turn myself off. So it's all yours, Bob. Next speaker.
So, I don't believe there will be an introduction, so I'll jump in. So, good afternoon, everyone, or good evening, anyone who's listening from this side of the Atlantic. My name is David Ponting. I'm Principal Scientist at LASA Limited, and I am going to speak on why nitrosamine potencies vary so widely, because they vary across 4 orders of magnitude. So why might this be the case? And it all comes down to the chemical structure. So before I really dig into this, we should have a short digression into what structure-activity relationships actually are. SAR is ultimately the association of chemical structure with a behavior. It's a more general term than toxicology, though it is in toxicology that we're talking about it. And from that perspective, you can have impacts of a substructural feature. These may impact typically chemical reactivity. So in the case of nitrosamines, you've got metabolic activation, you've got detoxification reactions, and of course, you've got the reaction with DNA and proteins. And this table at right just gives you an idea of the effect of the alkylating potential for some fairly small structural changes, methyl, ethyl, propyl, butyl. You can also have SAR of the shape of the entire structure. This is more important when you're considering an active site binding system or binding to a receptor, and this for non-covalent toxicity, less relevant perhaps for nitrosamines. And the properties of the entire structure affect ADME. Does it penetrate the cell membrane? Does it bind to plasma protein? Which organs are you exposed to? In the case of nitrosamines, probably the liver, but many others are also impacted. And from a metabolism point of view, is it polar enough to be excreted unchanged? An SAR can be quantitative, so a mathematical model, or based on expert assessment. And so for nitrosamines, the SAR ultimately comes down to the impact of the structural features on the stages of this mechanism. One side of the nitroso group must be able to be metabolized. The other side must be able to form persistent DNA adduct. And so within this, we need to consider pharmacokinetics, the metabolic potential, DNA binding potential, DNA repair mechanisms. As Bob's mentioned, a variety of metabolic enzymes are relevant for nitrosamines, CYP2E1 for the smallest, but the moment you move away from those tiny nitrosamines, CYP2E1 has a tiny active site. You start using a lot of other isoforms. R2 affects the rate of DNA alkylation and deactivation with water, but I'll mainly be talking about the impacts of changes in R1. So firstly, pharmacokinetics, and to really hammer this point home, the classic example will be the carboxylic acid containing nitrosamines. The majority of those that we have data for are non-carcinogenic, and indeed nitrosoproline has been used as a biomarker. It's been excreted quantitatively. There are exceptions. Nitrosomethyl carboxypropylamine, it's a bladder carcinogen, not liver. So even though it is carcinogenic, there's different behavior. They can be sphincteronic in physical conditions, and there's a specific CYP isoform that prefers to metabolize acid-containing compounds and has specific steric requirements related to the acid binding. There's a specific distance between the carboxylic acid and the active, where the carboxylic acid binds and the active site. But of course, they're already polar. The carboxylic acid group, particularly when it's deprotonated in physiological conditions, makes them really polar. So they might skip phase 1 metabolism. And if you skip phase 1 metabolism, you don't expose your nitroso group to P450s. A really good example of this is the compounds at bottom right. Nitrosoglycine is negative for Ames and Kark. If you put the methyl group on, it becomes effectively a false positive, Ames positive, Kark negative. And this may be because the bacterial systems in the Ames are less effective at demethylation than a human or an experimental animal. Whereas in lab animals, you demethylate in preference to alpha hydroxylation. So the next thing we need to look at is the steric hindrance around the nitroso group. Metabolic activation by a P450 is a chemical reaction. It's got some fairly strict steric requirements. Don't forget that what you need to do is bring your alpha carbons of your nitrosamine really close to a large flat plane, because that's the heme group. And so anything that inhibits the approach of the alpha carbon to this flat plane really dramatically reduces the rate of alpha hydroxylation. So once you remove CH2 groups, at best, you're a weak carcinogen. Some are negative. The big exception to be aware of in this data set, if you look at this figure from Cross and Ponting, is the dinitroso compound, which has got the unhindered nitroso group. So when you're doing your SAR, just because the molecule has a hindered nitroso, you need to be aware that it might also have the unhindered nitroso, and that unhindered nitroso is liable to be the cause of the relatively high potency of this compound. what about electronics? So in terms of electronic effects, there are a number of ketones in the data set. Ketones are a fairly weak electron withdrawing group in the grand scheme of things when you compare them to trifluoroethyl or to, for example, a nitrile or something like that. And the enol tautomer that you have for them that's not necessarily available to all electron withdrawing groups, but is to ketones, promotes the acidity. If you look at the figure at top right, you've got the enol tautomerization, and that makes the alpha hydrogen more labile. At least for compounds derived from nitrosopropylamine, so NOPA in the middle, there is an alternate route to the diazonium available, always forms the potent methyl diazonium, as described for the minor activation mechanism nitrosodipropylamine here. But you don't just get conjugation with an electron withdrawing group. You can also get conjugation with an electronically neutral system. So if you look at the transition state shown in the middle of the bottom figure, you have essentially a 3-center electronic orbital, which dramatically stabilizes that transition state. I think you get something like a 10 kcal per mole effect on bond dissociation energy for an allyl group versus a saturated system. And that makes the allylic and other systems much more reactive. If you look at the overall carcinogenic potential, compare the tetrahydropyridine with nitrosopiperidine, there's substantial difference in potency there. I should take a slight diversion here into hydroxylated nitrosamines. Those who've read some of my recent papers will note that I said that they have no overall effect on potency. And this is because there are competing effects. Hydroxyl groups increase polarity, so you've got arguments from pharmacokinetics like the acids. Beta hydroxy groups could be oxidized to beta ketones, which increase potency. Terminal hydroxy groups can be oxidized to acids, so back to the pharmacokinetic argument. And overall, there wasn't much apparent effect on potency. However, I've gone back into this data recently. And if you look at the beta hydroxys and separate out from them the beta gamma dihydroxys, you suddenly see features that are significant. We're not precisely sure why this is, but, and it might be specific to the 2,3-hydroxypropyl groups, which come from one specific set of carcinogenicity studies and are responsible for most of the potent nitrosamines that match the OCCN double bond O feature that we had used to describe the beta hydroxy. We now have to say beta without the beta gamma dihydroxy, and this really reinforces understanding your data. So here is an updated version of the figure from Thomas et al., where we've managed to put a lot more nitrosamines correctly into the low potency bucket by making this change without having any more mispredictions. So that's what happens when you've got a dialkyl nitrosamine. What about a nitrosated aromatic amine? Now, these you can't activate on the aromatic group, full stop. The activation must occur on the other side of the molecule. Interestingly, nitrosodiphenylamine is positive, but it's extremely weak, and there are some proposed alternative mechanisms out there. It's not really worth considering in the cohort of concern, lacking its alpha hydrogen as it does. And of course, you form a different electrophile, this phenyldiazonium, which has different reactivity patterns with DNA. And it may depend on the electronic substitution on the aromatic ring, as shown at the bottom. As you increase the electron withdrawing nature of the ring, you decrease the carcinogenic potency. Cyclic nitrosamines bring their own complexities. The ring systems can lead to decreased activity, but as I said, the tetrahydropyridine is extremely potent. And ultimately, some of the reasons for these differences, you've got a different metabolic profile available. you might form an oxonium cation as your electrophile if the aldehyde kicks out nitrogen. And this might be your ultimate alkylating agent, not a free cation or the diazonium. You've also got constrained conformation, which gives you an increase in effective steric hindrance. This is comparable to boat chair conformation or locking that many may have studied in undergraduate chemistry. Small rings also seem to be less potent than larger ones, and this may again be because you've inhibited the ability of the molecule to arrange in such a way that it forms the correct transition state for this cyclic intermediate, which releases the aldehyde and the diazohydroxide. And the more rigid, smaller systems might not be able to rearrange correctly. Nitrosoamides can also be nitrosated. Some can be potent carcinogens, but they have no need of metabolic activation, and the SAR depends on the nature of the alkyl side. The amide, or nitrosamide, is cleaved in physiological conditions without the need for metabolic activation. So you go straight to your diazonium and your DNA alkylation step. It gets even weirder. There's a whole bunch of other N-nitroso compounds which can't form diazonium ions. So these are things like nitrosated pyrrole type nitrogens, where you're not going to break the aromatic ring apart. Nitrosated primary amines, which are unstable. Heterosubstituted amines, so if you've got an alkyl on one side, you can theoretically oxidize it there, but then you don't have a carbon-nitrogen bond left, so you can't have a diazonium leaving group. All of these, it could be argued, are probably outside the cohort of concern. I've mentioned DNA reactivity and repair, and in the interest of time, I would highly recommend reading the Wenzel et al. And Ferrer and Chrisman et al. Papers listed here. But just to note that there are SAR impacts of the different structural features for these. And just before I'm kicked off the stage, we have mentioned that there's lots of NDSRIs and they have a fairly different chemical space to the small molecules. And LASA have host and act as honest broker for a data sharing initiative about complex nitrosamines, for which we're currently preparing the third release. This has 150 Ames results on over 100 compounds and in vitro data from other assays. And with this information, we're going to be able to look at any systematic SAR changes between small and complex nitrosamines. Are there any changes? Are there any differences? More or less genotoxic? How can we do the extrapolation? And we're going to get all of this information out into the public domain as quickly as we can. Obviously, if anyone has data on complex nitrosamines, do get in touch. We also have the nitrites and excipients data sharing project, where of course, the nitrite content is critical, but can vary between suppliers. And so this has over 800 records on 84 different excipients for nitrite content. And this information was published last year in Bertzel et al., and the data set continues to evolve. And so with that, I'd like to thank an increasing list of colleagues, both at LASA Limited and across industry, academia, and regulators, and you for your attention. And hand over to Naomi, I believe.
That's right. Thank you, David. And here we go. Here are my slides. Hopefully everyone can see that. Thank you for the opportunity to speak today. It's my pleasure to speak to you on the topic of structure-activity relationships to inform setting acceptable intakes for nitrosamine impurities. My name is Naomi Kruhlak, and I am the scientific lead for FDA CDER's Computational Toxicology Consultation Service. This is an outline of what I'll be covering. I'm going to start just with a quick recap on some of the terminology around quantitative structure-activity relationships. And then I'll give a little bit of background on FDA's published guidance on the control of nitrosamine impurities in human drugs, which provides for the use of surrogates for setting AI limits. But I'd also like to touch on some of the challenges associated with setting limits, specifically for nitrosamine drug substance-related impurities, or NDSRIs. And then the majority of my time will be devoted to covering some of the research that's been going on at FDA CDER, some of the earlier work that we were involved in developing a local similarity method to more objectively identify potential surrogates for nitrosamine impurities. And then our more recent work looking at predicting carcinogenic potency using weighted structural features using a structural fingerprinting method. And then I'll finish up with some future directions. So I have a brief cap on QSAR terminology. So QSAR stands for Quantitative Structure-Activity Relationship. And as David mentioned, it's a type of modeling that identifies associations between attributes of chemical structure and biological activity. And that activity really could be any kind of biological activity. But in the context of this presentation, I'm going to be talking about mutagenicity and also carcinogenic potency. It works under the general assumption that similar molecules exhibit similar chemical and biological properties, and therefore that toxicity can be explained by chemical structure. QSAR and SAR collectively are often referred to with the Q in parentheses, but the distinction here is that QSAR is a quantitative relationship that's often developed through machine learning, sometimes referred to as statistical-based modeling, whereas SAR is a qualitative relationship that's usually human expert derived, and it can be encoded into expert rule-based models. QSAR models can be used to make a prediction of a chemical's toxicity based solely on the chemical structure of the molecule of interest, and typically it takes into consideration multiple structural aspects and contributing features in making that prediction. Read across is a manual structure-activity relationship approach where we would use a structurally similar analog to our data poor target molecule and then use the data for that similar analog or surrogate molecule to then read across to our target molecule, such as in the example that I've shown here on the right-hand side, where data for NPIP could be used to read across to the NDSRI shown below. FDA has published guidance on the control of nitrosamine impurities in human drugs back in 2020, and it was revised in February of 2021. And within this guidance, there's an appendix that describes how a human acceptable intake limit can be calculated by linear extrapolation from a rodent TD50 value. And that TD50 value represents the dose at which 50% of the animals in that long-term repeat dose carcinogenicity study exhibit tumors. So we can convert that TD50, which represents the 1 in 2 tumor incidence in rats, to an AI, which represents a 1 in 100,000 excess cancer risk in humans. And the lower the TD50 value or the AI, the higher or the greater the carcinogenic potency. TD50 values are available for about 140 nitrosamines reported in the carcinogenic potency database, sometimes referred to as the GOLD database, but this database is no longer being maintained. It was retired some years ago, but the data are still available through the LASA carcinogenicity database, where they present the original CPDB TD50s alongside a recalculated LASA value using a more standardized method for that calculation. Most of the data that we have are for small molecule nitrosamines, with potency ranging over 4 orders of magnitude. In fact, some are reported as non-carcinogenic, but many of the studies are not highly robust, and that can be a significant limiting factor. So in the absence of robust empirical carcinogenicity data for an impurity, an AI can then be calculated by using that read-across approach from a structurally related analog or surrogate molecule by taking the TD50 value for that molecule and applying it to the untested impurity. As Andre had mentioned this morning, nitrosamine impurities really can range from being very small, simple molecules, often monofunctionalized molecules, to being these very large drug-like nitrosated molecules that have lots of different structural features and functional groups. And those are more complex structures, or what we refer to as NDSRIs typically. The challenge with NDSRIs, though, is that we usually have no mutagenicity or carcinogenicity data available for those structures, and we're not expecting to be generating new carcinogenicity data. Also, those structures are often unique to the drug substance. So if we think about the chemical space of all possible drug molecules and the potential for those to then be nitrosated and form NDSRIs, we're talking about a very broad area of chemical space. And for risk assessment, we would need to determine the mutagenic potential and the carcinogenic potency for those molecules in the absence of actual experimental data, and that's very challenging. So that really led to some of the research efforts at FDA CDER. In our initial work, we focused on ways to more systematically identify an appropriate surrogate that could be used to read across to our untested NDSRI by focusing more on local similarity around the nitroso group environment rather than global similarity, which is typically the method that was available at that time. So in order to do this, we worked with Multicase Incorporated under a research collaboration agreement to help develop a structural fingerprint that could allow us to capture the chemical structural information up to 5 bonds out from that N-nitroso group for a set of molecules. And that fingerprint could then be calculated for the untested molecule and then compared to other molecules in our database for which we already had data to find the ones that it best matched to help us really more objectively identify appropriate surrogates. So this has some utility, but the challenge is that we're still limited by the fact that there's a pretty limited number of surrogate molecules available that actually have robust enough data to support an AI calculation. And in fact, some areas of chemical space may have a lot of studies, but actually none of them are sufficiently robust. And nitrosated piperazines come to mind as an example of that. So we really need to look for a prediction method that's not reliant upon having data for a single compound. And furthermore, it would be ideal to have a method that's less subjective in the sense that for surrogate selection, there's still cases where there's some concern about whether that surrogate is sufficiently similar to actually be able to use for read across purposes. So how similar does it need to be in order for it to be acceptable? So to move away from that, there was an interest in developing new prediction methods that could consider multiple structure, metabolic, and physical chemical factors in a molecule. This would give us the ability to leverage a larger body of training data for the identification of SAR patterns. And that's helpful because it means we're not relying on single studies being highly robust, but in fact, we can tolerate a lower degree of robustness because we have a larger number of observations to work with. We also have the ability to learn from relative carcinogenic potency data, even if we actually don't have TD50 values available for those studies. For example, if we have a pair of compounds that are tested in a carcinogenicity study under the same conditions, but one causes a higher rate of tumors than the other, we can conclude that there's something about that compound that's more potent, and we can start to look at the structural features that might be contributing factors, even though we actually don't have a TD50 value to learn from. We also, by looking at the problem more holistically, have the ability to integrate chemical reactivity data or other ADME considerations, which you recognize as being very important parameters. And that was something that David had mentioned in his presentation. So we're fortunate that a significant body of work has already been done and published by many scientists in the area for SAR patterns for nitrosamine. So can we learn from that? Can we use that information? This just highlights a few of the leading publications that we've seen. And this highlights some of the SAR trends that we're already aware of. So this led to a collaboration with other international drug regulatory authorities through the Nitrosamines International Technical Working Group, or NITWG, with the goal of coming up with a way of setting an AI by predicting potency using a combination of activating and deactivating structural features in the nitrosamine. And we found that categorical prediction shows good promise. To facilitate this work, we created a data set of 84 nitrosamines with rat TD50s from the CPDB LCDB, and also with relative potency classifications as published by Rao et al. In his 1979 publication. The molecular weight range of these molecules is notably low. You know, we're talking about small molecules here, ranging from 74 up to 278. But this is what we have data with, so it's just really a limitation of what is available to work with. And that data set, I should point out, corresponds to 90 individual N-nitroso group environments, because some of those molecules have 2 groups. And that's important to point out, because potentially the ability for those 2 groups to be metabolically activated could be different because they're in different chemical environments within the molecule. So that's something we need to consider separately when we're looking at applying SAR patterns. So we assigned relative weights to a range of activating and deactivating features based on SAR trends that have been published in the scientific literature, SAR trends that we observed in our data set, and also mechanistic rationale, just thinking about relative reactivity from the standpoint of what we know about chemistry and biology. And then we assess the prevalence of those features using a molecular fingerprinting method across the set of 90 N-nitroso groups in our small molecule data set. And then for comparative purposes, we applied that same set of fingerprints to a set of several hundred drugs that are marketed in the US that have nitrosatable amines and could hypothetically form an NDSRI. So this focused on molecules that have secondary amine centers or tertiary dimethylamine centers. And each N-nitroso center was evaluated separately for the purposes of this exercise. And just to kind of give you an idea of the difference in the molecular weight range when we look at these hypothetical NDSRIs, the drugs that we had in our data set have a very different distribution in terms of their molecular weight compared to that small molecule data set that we actually have TD50 values for. And notably, the majority of the drugs that we're looking at fall into that 250 to 500 grams per mole range. So we started by looking at the alpha hydrogen environment, specifically looking at the number of alpha hydrogens available on each side of that N-nitroso group and their distribution. And so if we think of all the possible combinations we could have, they're shown here in this table on the right-hand side. And the far right columns show you the prevalence of those features, those substructures in the small molecule data set and then also in the drug data set. I think what's quite striking is for the small molecule data set, the majority of the alpha hydrogen environments fall into the high potency category, meaning they're sterically unhindered and much more amenable to metabolic activation, in contrast to the drug data set, where we see that only about a third of the molecules actually would fall into that high potency range, suggesting that many of these NDSRIs are going to have lower potency. Similarly, we see for the drug data set that 14% of the examples actually have no alpha hydrogens, and this would suggest that the alpha hydrogen or alpha hydroxylation mechanism is not going to be available. Moving on to activating and deactivating features, there's a significant number of deactivating features that have been described in the literature. And some of the more common features that show up in our data sets, which have a very strong deactivating effect, include a tertiary alpha carbon on one side of the N-nitroso group, which shows up in about 6% of the drug data set, and the carboxylic acid group, which David also mentioned, which has the potential to increase clearance and therefore lead to lower potency. And that shows up in about 13% of the drug data set. So these are pretty significant numbers of NDSRIs that have these strong deactivating features. Electron withdrawing groups on either one or both sides are also prevalent in about 10% of examples in the drug data set. And as an extension of that, the hydroxyl group in the beta position, which is expected to have a deactivating effect, shows up in about 20% of the drug data set. The ring structures ranging from 5 to 7 membered rings map out to about 17% of the drug data set. And this is interesting to note just because we recognize that these types of environments are likely to go through that different mechanism where the following cleavage of the nitrogen carbon bond, we can then have recyclization to form a cyclic electrophilic species. And then lastly, another feature that we defined through our work was where we have 2 chains of at least 5 atoms on either side of that N-nitroso group. And that's really to reflect the position of that N-nitroso group in the molecule, where that molecule has the N-nitroso group kind of in the middle, buried within these other flexible parts of the of the molecule. It's likely to be less amenable to metabolic activation compared to when that N-nitroso group is on one end of the molecule. And so we would expect that to have a deactivating effect on carcinogenic potency. And this feature shows up in about 1/4 of the NDSRI data set. We have a couple of examples of features that are associated with an increase in carcinogenic potency. The most prevalent here is the benzylic-like feature, which shows up in about 10% of molecules. So with respect to future directions, the combined weight of features that we've looked at through our study may be used to generate a prediction of carcinogenic potency for an untested NDSRI. So this gives us the ability to consider the effect of multiple features in the molecule, which is important for complex molecules such as NDSRIs. It allows us to leverage a larger body of available carcinogenicity data than a surrogate analysis where we're dependent on that single robust study or that robust data for a single compound. And we can refine these features and weights as new testing data become available over time to improve our ability to make a prediction. And I see this as being similar to the evolution that we've seen in ICHM7 QSAR models that are used to predict the mutagenicity of other classes of impurities. We can also, as time goes on, we can integrate additional data types and chemical and biological considerations into our predictive approach as they emerge. And this could include quantum mechanical descriptors or metabolism data. And then furthermore, structural fingerprinting of NDSRIs can help inform future testing and modeling. We recognize that NDSRI chemical space differs substantially from the chemical space of the small molecule nitrosamines for which we currently have data. And so we can use these fingerprinting exercises to identify where there are data gaps to then inform the selection of new compounds for testing initiatives so we can build out our knowledge base and build out our model. And then as those new data and knowledge are generated, we can then incorporate it into our model to improve its performance over time, to improve its prediction accuracy, as well as its structural coverage. And with that, I would like to acknowledge the many individuals who contributed to this work at FDA from the Pharm Tox groups and also the computational toxicology group, and also our international collaborators through the TWG, and specifically acknowledging the contributions of my co-lead on this work, Mariana Schmidt from EMA. Thank you for your attention.
Yeah, so I'm going to talk about hydrochlorothiazide, and I'm going to talk about some specific aspects of investigations we've carried out that suggest that, in regards at least to the data that we originally collected, particularly around the Ames test, that there is something of an anomaly associated with that, which ultimately leads us into a narrative around a series of investigations we ultimately did to really determine whether or not nitrosamine, the nitrosamine of hydrochlorothiazide itself should actually belong to the cohort of concern. So, a bit of background. I mean, hydrochlorothiazide is a diuretic medication. It's been used since the 1950s, primarily for hypertension, but also as well for edema. It's one of the most commonly prescribed drugs that is used, both as a single agent and often in combination with other APIs, particularly the beta blockers, the ACE inhibitors, and the angiotensin receptor blockers as well. So it's a very, very commonly prescribed drug. In fact, it's the second most commonly prescribed drug. Typically is a combination product in the US with nearly 40 million prescriptions and nearly 10 million patients, and that's the US alone. So it's an important drug. The but is, and relating to the nitrosamine situation, is that hydrochlorothiazide is susceptible to nitrosation. It's an anilinic secondary amine, and that is a very weak base, and that makes it very, very easy to actually nitrosate it. So with that in mind, and realizing from very early on in terms of data we were collecting at an industry level and as regards to levels, that we realized that this was going to be a potential concern, particularly because of that facile nature and the fact that we were clearly seeing the nitrosation of hydrochlorothiazide. So the obvious thing that was done was an Ames test was carried out. And as far as the Ames test was concerned, it was Ames positive. But the key thing is, and this is where the twist in the tail starts to happen, is that the profile was not typical for a nitrosamine. It certainly would indicate the Ames profile that the situation was more complex than we'd originally anticipated. So what we started to do then was a series of investigations really to look at the nitrosamine of hydrochlorothiazide. And one of the first things that we clearly demonstrated and could see was that the nitrosamine itself, although facile to form, was equally extremely unstable under physiological pH. And so the question as regards that is, so what? What does that actually mean? Well, I've already said we carried out an Ames test. One was carried out within AstraZeneca, our own organization, and it was Ames positive, and we could get repeatable results. But the interesting thing that we saw was that we were seeing activity, we were seeing a positive response both with and without metabolic activation. And that is not consistent with the anticipated profile. I don't need to go over that profile again. You've heard it from a number of other people already in this over the last day or so that we've been talking about this. So I don't want to repeat that aspect of it. But looking again at this from the context of what is this telling us? Well, I mean, the fact that we're getting a positive response without activation in TA100 and E. coli strains is suggestive that there is something else present within that system, that there's a complexity associated with this, particularly in terms of the fact that you appear to have a direct acting, perhaps a non-nitrosamine alkylating agent present in the system. So the obvious question for us to investigate was really, what is it and how is it formed? Well, I've already said that nitrosyl hydrochlorothiazide is really unstable under conditions that are analogous to the Ames test. So if you look at it in the context of the Ames test with pre-incubation, we pre-incubate 37 degrees for 2 hours. But look at the study that was done effectively at neutral pH. Just from a chromatography point of view, it is very, very apparent that certainly over that 2-hour period, that in terms of the species that would be present in the Ames test, there may well be some nitrosyl hydrochlorothiazide present, but in reality, most, if not all of it, would have degraded, and that there were clearly, therefore, other species present within the Ames test. So what is it degrading to? Well, this was the start of the investigation. We looked to identify what some of these particular species were that we were generating. So we would do what you would expect that we would do. We carried out an investigation looking at identifying what each of these components were. And although that shows several structures here, the really important ones to highlight in terms of what we actually saw in reality in terms of the substantive levels were we saw what we referred to as impurity A. We also saw something referred to as impurity D. And as part of this, as part of that physical degradation, we also saw the formation of formaldehyde as well. So formaldehyde, of course, is something that is a direct acting mutagen. And so what actually happens is, and you can track this in the within those, under those conditions, is that nitrosyl hydrochlorothiazide degrades rapidly to form these two major impurities as well as formaldehyde. So the next step was to think about, well, mechanistically, what was going on. And so a proposed degradation pathway of nitroso hydrochlorothiazide, particularly to the thiotrizine, was investigated. And the problem with this was, as far as this proposed mechanism was concerned, was that it suggested that the nitrosamine that we would be, the diazoniamine rather, that we'd be concerned about, particularly in terms of what would form in vivo, it could actually also form from a degradation pathway point of view. So we would still have, in theory at least, that risk in terms of the nitrosamine risk, or more specifically, the fact that we could form the specific diazonium species either as a metabolite or also potentially as a degradant as it decomposes to these other species. But further investigation has now realized that the diazonium ion is not formed. By generating authentic samples of the postulated species, including the diazonium ion, we've been able to identify more clearly, at least provide a very clear rationale for the basis of what actually is happening in terms of the degradation of regards to the nitrosamine of hydrochlorothiazide. And time doesn't permit me to be able to go through and explain this in a great deal of detail, but I can make some clear observations as regards that. So that hydrochlorothiazide, the diazonium that we would be so concerned about, is not observed during the decomposition of the nitrosamine of hydrochlorothiazide. Indeed, as part of this work, in order to be able to evaluate this, an actual authentic sample of the diazonium ion was actually prepared. It had to be prepared in acetonitrile solution under extremely acidic conditions, 12 equivalents of sulfuric acid. It's the only conditions in which it's reasonably stable. Clearly, you can see from that that such conditions are not physiologically relevant, and immediately in the presence of water, it rapidly transforms to other derivatives. Now, there are other diazonium species that can be generated as part of this. So there's a sulfonyl diazonium species, and that has been shown to be a key intermediate in the degradation. But the thing with that is that this is more analogous, that sulfonyl diazonium is more analogous to a sulfonic acid halide. And so really that should not be considered as part of the cohort of concern. And similarly, there was another diazonium as well. If you go back to the Ames test, I said it was complex. How do we explain this and deconvolute this? Well, what became apparent was that the Ames profile of the nitrosamine of hydrochlorothiazide was consistent, certainly in terms of a positive, with what we see from the point of view of the Ames profile of formaldehyde itself. We also tested in the Ames study the actual one of the other degradants, so we got an authentic sample of the trizine. That again is actually Ames positive, but this is again without activation. So it is completely different to the profile of a nitrosamine, and more obviously as well, it's not structurally similar to a diazonium. So what are the conclusions? Well, nitroso hydrochlorothiazide is extremely unstable, and the physiological pH, it generates the species I've described, an equimolar amount of formaldehyde, an aromatic amine, and a thiotrizine. Formaldehyde has a limit from a safety point of view. This is listed within ICHM7 of 10 mg a day. From the nitrosamine, it may form at a few hundred nanograms. There's no way you can generate anything like the level of formaldehyde that would be of concern. More importantly than anything is none of these belong to the cohort of concern, and they're not nitrosamines. So on the basis of that, at least in terms of the impurities that we do see, the impurity A and impurity D, they would effectively, in our view, be managed through under the TTC at 1.5 micrograms per day. And just to bring some symmetry and conclusion to all of this, mechanistically, the mechanisms show, and the studies show that we don't generate the diazonium ion that would be of concern. And this is mirrored in terms of the transgenic study, where we now have a clear negative as regards this. This fully corroborates the results and the finding of the mechanistic study. So having completed all of that work over the last 18 months, our conclusion at least, and the one that we propose now, is that nitroso hydrochlorothiazide is actually non-mutagenic. It belongs to class 5 and can be controlled to limits defined by Q3A, Q3B. So in my view, this work actually demonstrates that in terms of hydrochlorothiazide itself as a drug, that this is not a significant concern, particularly in the context of nitrosamines. So with that, I'll stop. And I'll thank you for your attention.
So I think it's a great opportunity to be able to present it, particularly at the end of this session, because lots of what I'm going to discuss is based on and consistency with the other topics that were already mentioned in the by previous speakers. And what I'll present to you today are several position papers that were recently put together by a group of scientists from the FPIA under the auspices of FPIA. And the reason for for working in such a manner is that it is very difficult many times to find a good surrogate to do read across for NDSRIs. And therefore, we needed to use a different holistic approach and try to do some type of SAR and mechanistic understanding in order to find an appropriate limit for these full classes of NDSRIs. So the disclaimer, of course, is all the opinions expressed here are mine and don't represent Teva's official opinions. So the position papers that were put together were for three different classes that several already were mentioned by Andy. These consist of the calcium channel blockers, the dipines, commonly known as dipines, the beta blockers, the OLOs and the beta agonists, and the ACE inhibitors, which are known as the PRILs. So we'll start with the dipines. What we see here is we have a very, very common scaffold amongst almost all of the these calcium channel blockers. And what we see is that the amino group of the dihydropyridine is a secondary amine, which can theoretically undergo nitrosation. And the nitrosamine that you would get is one that does not have any alpha hydrogens. Now, upon investigation of this compound, we tried to actually synthesize it, and we were unsuccessful. And what we did find was, and also based on this was actually some literature from the past that already mentioned this, under conditions of nitrosation, the dihydropyridine prefers to aromatize and it does not undergo nitrosation. Even though there are vendors that claim to be selling nitroso calcium channel blockers and nitroso amlodipine, for instance, we challenged this the vendors themselves, and we investigated their NMRs of the substance, and it was indeed not a nitrosamine. It was the aromatic species. So this is more of a quality type of approach. And of course, when you cannot synthesize the nitrosamine, there's no formation, so there's no risk. But even if theoretically, if it would have formed, again, it does not have any alpha hydrogen, so it would not be able to undergo the alpha hydroxylation, and therefore we could essentially exclude these from the cohort of concern. But as I said, based on the quality aspect, this compound, these class of nitrosamines don't even form. A larger group of compounds is the beta blockers and the beta agonists, which they also have a very common scaffold, and we can separate them to do different groups here. We see them in the magenta color as the common features of most of the most of the beta blockers. A couple are excluded from that. For instance, which you see on the bottom of this screen, which is the nitrosocarvedilol and the nebivolol for the beta blockers, or the vilanterol for the beta agonists. But all the rest of them really do have a very common feature where one of the alpha positions is substituted by a very bulky group, either an isopropyl or a tert-butyl group. That's on one side of the nitrosamine. On the other side of the nitrosamine, they all have the beta hydroxy feature, which was mentioned several times both by David and Naomi, that in it being a deactivating feature. So we have here altogether multiple deactivating features: the bulkiness, the tert-butyl and isopropyl groups, and the alpha hydroxy, the beta hydroxy, and some other previous studies. For instance, there was a study from Martinelli in 1994 that showed that clastogenicity was considerably less potent in comparison to NDMA. We know that as we said, the beta hydroxy is a potency reducing element. The isopropyl group and the tert-butyl group, they're all very bulky. They're going to considerably reduce the potency. The outliers, I call them outliers, those that don't have that feature, that don't have the bulkiness at the alpha position, they still do have other features which are deactivating. For instance, the beta hydroxy, as Naomi indicated, in it being even a stronger deactivating feature. And the comparison of that would be Andella and nitrosodiethanolamine, which is a very weak carcinogen. And also by doing an analysis of the metabolic pathways of these particular beta blockers, there's probably other alternative pathways rather than the beta of the alpha hydroxylation. The Ames tests of this whole class, the data is somewhat conflicting. Some are positive, some are negative in using modified protocols. So it's difficult to rely on the data coming from the Ames of this whole class. However, several TGR studies are currently undergoing, which will hopefully elucidate this whole class. And the expectation is that the deactivating features are going to show them to be either non-mutagenic altogether, or if at all, they'll be with weak potency. And here again, with the overall weight of evidence, indicates that this class, the class of both of these, the beta blockers and beta agonists, can be excluded from the cohort of concern. The third class of compounds is the ACE inhibitors. Here again, we have a very common scaffold amongst almost all of the prils, these ACE inhibitors. And here we have even a more complex structure around the nitrosamine, a more sterically hindered structure, where both alpha positions, there's only one hydrogen. We have on one side of them all, there's an ester group. On the other side, there's an amide group at the beta positions. And all of them have at least one, if not two, free carboxylic acids. And all these features together are very highly deactivating. Just looking at the three-dimensional structure of an example of the nitrosoenalapril here on top, we see that the alpha positions are extremely sterically hindered. The access to them in order to undergo the alpha hydroxylation are very, very difficult. And naturally, quantum chemical calculations showed that it would be very improbable that hydroxylation would occur at either of the alpha positions. But nonetheless, even if we would theoretically, they would undergo that hydroxylation and form either of the diazoniums, we can see here that the diazoniums themselves, diazonium analogs, would be also very sterically hindered by the other side of the compound, and it would be very difficult for DNA to actually fit into these grooves and interact with this diazonium. So altogether, for the prils, we see that there's considerable steric hindrance on both alpha positions, which prohibit alpha hydroxylation. As we said, the quantum chemical calculations predict this to be a very low probability of undergoing the alpha hydroxylation. The steric hindrance around the putative diazonium ions would prohibit DNA binding. The free carboxylate is a very deactivating feature, as mentioned earlier, both by David and Naomi. Here, actually, all of the compounds that were already tested and that are reported in the LASA complex nitrosamine data sharing project that David described earlier, they're all negative, with a few that are positive but without S9, a very few, I think one or two of them that are positive without S9. So there's none that are actually positive with S9. That's a strong indication that this whole class is probably non-mutagenic. Several in vivo mutagenicity studies, both TGR and duplex sequencing, are undergoing, and several have already been conducted. And those that have concluded have already shown them to be totally negative, both in the TGR and the duplex sequencing. Here, the overall weight of evidence is even stronger than within the beta blockers, and they should at the least be excluded from the cohort of concern, if not altogether considered as non-mutagens that can be controlled at the ICHQ3A and B. So to conclude, we have three different classes. One of them is more of, as we said, a quality issue, probably no risk of formation. And therefore, they should not be considered altogether. There's no need to test them altogether. You can't even prepare a reference standard for them. The beta blockers and the beta agonists and the ACE inhibitors are all to be considered as very weak carcinogens, if not mutagenic altogether. And with that, I conclude, and thank you.
My thanks to all the speakers. We are a little bit behind on time. We have about 20 minutes, so it's a tall order to get through all of these topics in depth. I'll ask Dr. Sruthi King to please come to the front, and I want to make sure that our speakers can also hear us online.
Yeah, we can.
Yep, okay.
Yes.
Okay, thanks. I'd like to break the panel discussion into two different areas. The first focusing on predictions and estimates, the second on experimentation. Okay. So first question going to Dr. Ponting and Dr. Kruhlak. If we're implementing in silico models for prediction of enhanced Ames mutagenicity and carcinogenic potency, do we need to keep testing these nitrosamines in experimental assays for these endpoints? And I'm going to reframe that by saying, how close are we to solid, reliable predictions, the likes of which you've discussed in your presentations?
So I think the long-term view, no, we don't need to do the experimental tests. We will ultimately be able to have solid in silico predictions, not just a binary call of, is it likely to be carcinogenic or mutagenic? Because a lot of them are likely to be mutagenic. But a potency prediction and a refined classification like that, which hopefully can then be built back into an M7 framework. So you'll end up being able to say, yes, it's cohort because it's an heteroso group, but we don't expect it to be super potent, so we can control it to TTC. Even though it's not TTC doesn't apply, that might be a useful line to draw. In the shorter term, I think once we have a readout from Bob's work and the Hesse work about the Ames activity, and we can then be really confident, and we've got the extrapolations of the small molecule into the complex and a little bit of sort of essentially synergistic work amongst the different modeling teams, I think we'll be pretty close.
And I would agree with that. I think in the near term, we definitely do need some additional data. Testing is going to be around for some time yet. And we can certainly benefit from that for the modeling activities. But the ideal goal would be to be in a similar place to where we are under M7 for other types of mutagenic impurities, where we can really rely confidently on these models. If we think of the timing for that, M7 was finalized in 2014. So we're 10 years out, and we're very confident in using those models instead of an Ames assay. Maybe we're looking at a similar timeline, but I think in the near term, we can definitely benefit from having new data available and to belabor a point in a way that modelers can have access to it. So data sharing initiatives so that everybody has access to it to learn from it for the benefit of the broader scientific community.
Well, Dr. Kruhlak, it's as if you read my second follow-on question. Because it's all about data access then if we're looking to build the models. And so now that, we're in a large group, we're creating data, how can we ensure that the data that we're producing is accessible to in silico modelers? What should we be keeping in mind? Is it publish the results? Is it consortiums and being involved in those?
So I think David could certainly speak to this because LASA has been working very intensively to serve as an honest broker for a lot of these studies that have been generated so they can be shared more broadly. I think there's still some work to do there to make it available to a larger body of people. Not everyone has access to LASA's databases. Publication certainly helps. So getting the word out in the published literature is certainly a way to make sure that everyone has access to it.
So we're working with the members of the consortium to enable regulatory access to the database, and yes, Naomi says publications. The proprietary data within the database is such that to throw the database open generally is impractical. However, the information from that database, the knowledge about SAR, can be released without breaching company proprietary information. And that's what we'll be working towards, certainly in the second half of this year. I know that there's other people interested in SAR amongst the members of the database. And we'll be hopefully looking to pull out some really valuable learnings and share them as widely and as quickly as we can.
Thank you. And I do want to invite the audience in the room to please come to a microphone at any point where you're compelled to make a comment.
I thank you so much, Andy, Rafi, David, all of you. And I was going to actually ask this question about the sharing of the data. And I do understand we talk of consortiums and everything, but the pharmaceutical industry is not a nonprofit organization, neither is LASA from the last time I heard. So it is going to be difficult. And there are, I know there are bigger companies who are working diligently, but I would also think of the smaller groups who, not just generic, even new drug, who have, say, 2 drug products or 3 drug products, and it's very difficult for them to access. And I think this is a safety issue. I personally feel that this is FDA's responsibility to communicate, and I know it's difficult. We are a litigious society, putting information here and there cause problem. But I would strongly, recommend and also request to the agency to see how they can probably share this more with the industry. But I had another question. I did not hear anything about molecular weight adjustment and less than lifetime. I know agency doesn't like either of them, but I would like the panel to opine. And Naomi, especially, I saw your data. I would like to know what your understanding is, because ICH M7 allows it for others. And just because nitrosamines are cohort of concern doesn't mean they are out of the world. They are still very much part of M7. And this is something we should probably have a little more understanding of, and it will greatly benefit the industry.
So Alok, on the particular point of the LTL, that did come up in some of the discussions at the recent FDA HESI meeting, and others will probably want to comment on this as well. But what I heard was that in terms of the principle of using the LTL approach as far as nitrosamines, that there was a tacit recognition that LTL principles could apply to nitrosamines, but at the moment we only had a limited data set because the only data that would support this was based on NDEA. And the opinion, the view that I took, and I thought I heard in this meeting was that there was a call really to do some further research in this area to see if there's other examples that could be studied in this way, maybe some of the other carcinogenicity studies, in a way that we could support that data with further data sets in regards to this. But it was the first time I'd heard that we were moving towards at least a tacit acceptance of the principles that we could apply LTL principles to nitrosamines, albeit with a caveat of needing more data. But I mean, David, Naomi, or Bob, you were all in the same discussions. That's the interpretation that I put on it when I heard it. But I'd be interested in others' views on that one.
Yeah, for those few compounds that we do have enough data, it's very much worth doing a similar exercise to what we did with NDEA a couple of years ago, just to confirm it. In terms of molecular weight adjustment, I think there's significant value in it. Ultimately, this is a stoichiometric process, the DNA adduct formation. One molecule of nitrosamine, one adduct, 1 mole of nitrosamine molecules, 1 mole of adducts. And so it would make scientific sense to have molecular weight adjustments applied. Obviously, the final limits will still need to be expressed in mass units, but I see no inherent theoretical barrier to using a molar way to scale those, particularly if your read-across analog is significantly smaller than your NDSRI. Just as a point of order before I let go of the microphone, LASA and Limit are a non-profit organization, which is what enables our honest broker status amongst these many data sharing projects.
Okay, are there any final comments on less than lifetime or the vector of molecular weight?
Just one very quick one. In the HESI meeting, Joel Burke, you did mention that the less than lifetime concept was actually developed mainly on nitrosamines. So that in itself is a good justification to use the concept.
Okay, thank you. Our time is relatively short, so I will move further down. Oh, excuse me. Yes, in the room.
Hi, sorry, I have one question. A lot of regulatory agencies have already published AIs for a lot of NDSRIs, like EMA, FDA, Health Canada. But as we're developing all these new tools, in particular the ones Naomi presented, and we're getting in vivo data, will the FDA potentially work with other agencies to go back and look at some of those AIs that they maybe have calculated based on a simple read across maybe a year or two ago, and maybe revisit that calculation, particularly if it shows it to be less carcinogenic than originally thought? Would the agency consider revising those limits based on additional data and improved modeling?
Thank you for your question. I think it's certainly something we should take a look at. I can't say that those limits will be revised, but I think it is important for us to look at the data that were generated in order to inform those higher limits. So at this time, there are no plans to revise any limits, but I'm not going to say that we won't consider any information that's submitted to application.
So just a very quick comment and reflection on that. And I think this is an area that shows perhaps the need for further collaboration between different regulatory authorities, because we had a meeting last week with the nitrosamines expert group from the European Medicines Agency from a safety perspective. And actually, I mean, in terms of the things that they talked about, they did proactively talk about going back and reevaluating some of the current NDSRI AIs, and actually potentially in an update to the Q&A document, actually publishing some revised limits for retrospective revision of some of the NDSRIs, for which we have it already in AI, but we now have new data that would suggest that should be modified. So I can't comment in terms of the discussions that happen between regulators, but I mean, certainly would seem in this particular case, maybe the European Medicines Agency is slightly ahead in that discussion.
I guess I would just add, thank you for sharing that. Certainly the EMA is within their rights to pursue, the approaches that they wish to based on the information that we're seeing. Speaking for FDA, I think we'd have to look at that information first and then determine if a revision is necessary for limits that we've published.
Just a follow up to that. So I guess what's the agency's view on like the Big Blue study in rats and stuff? Would they accept that as a part of a justification for wider limits on some of the currently published limits on nitrosamines?
I mean, I think you've heard Dr. Heflich mention that FDA is also looking into investigating, studies in Big Blue rats. So I would say we're open to additional justification. Somebody mentioned this morning, we're learning as we're doing. So I think this is an area that science is rapidly growing. There's lots of new information coming in. However, I think we would need to see the information in order to make an informed recommendation. I know there are a lot of studies going on in multiple areas across multiple groups, but it's important that we actually see the data as regulators in order to make a decision on that application.
Understood. Thank you.
Okay, so I'm going to turn towards experimentation and a question for the group. What is the value of in vitro mammalian cell data in evaluating the toxicity of NDSRIs? Other than testing compounds for which the Ames test is inappropriate, how can these other in vitro tests be used?
Don't know, have we lost Bob?
Oh, no, I'm here.
So,
That was actually my question. And I mean, I have a view of that as far as my position is, but my sense is that the people who are conducting these studies in the pharmaceutical industry and the other areas of FDA aren't putting very much value on the in vitro mammalian cell studies. And I just point out that Emma has a big initiative to evaluate some of these endpoints. So there seems to be a disconnect there somewhat, and I just wondered whether anyone had any opinions on this other than myself.
I think just from a general point of view, a concern that I certainly would have would be if you look at a scenario where you have for instance, a name's negative with the revised protocol. It's now, we've got all the data that you're doing a fantastic job collating at the moment, Bob, and we agree on that protocol and we test something and it's Ames negative. If somebody then goes and does one of these other in vitro studies and gets a different endpoint, a different outcome, then what is really unclear to me is, well, how do you weigh up those, what in effect then would be two conflicting results? I mean, how would you balance that out? And in particular, how would that inform you ultimately in terms of relative potencies of potential AI, or even defining whether it's a concern or not? It's just the devil's in the detail of how you would interpret it when you've got this additional in vitro data, particularly when it's from an assay which is not directly itself a measurement of mutagenicity. I mean, it's more looking at things like, the micronucleus test and comet assay and DNA damage. So I'm just struggling a little bit to see how you would fit that all together in that jigsaw.
and towards that, if I could ask Dr. Heflich, thinking about the other in vitro assays that you've been conducting, I realize you've done extensive work using hamster S9 in the Ames assay, and really trying to make that the most sensitive assay. So when there's a positive with the hamster, certainly that's informative. And what I wonder is also, are these other in vitro assays generally showing a positive result that lines up? Is there unity between the Ames positives as well as in these other assays so far in your experience?
Well, without letting too many cats out of the bag, for the NDSRIs, they've lined up perfectly so far. In other words, something that's positive in the Ames assay, whether it be enhanced by hamster S9 or in one case is totally dependent on hamster S9, does appear to be positive in the human systems using human metabolic activation. So I think there may be some, if we're really ultimately concerned about human risk, I think that information is important to consider at least.
But that would be more of an academic exercise, and it would be
I don't think
nice
so.
to have.
Don't think so, Ralphie. But that may be where the guidances are written now. But I mean, they're written from a pragmatic standpoint, in that is where the data is. It's in rodent systems, and here we've got some human systems that we can look at. And we're ultimately giving these drugs to humans and not to rats and mice.
Absolutely, but so my intention was not to disregard the human data, but the human microsomal data, but to say that, and in line with what Andy was saying, are we going to actually have to test in both systems for every case? And I think that's the proof is in the pudding. And will we ultimately be able to rely just on the modified Ames, or are we going to have to do multiple studies? And where's the limit? Where are we going to stop? I mean, right now we know that even if we have a negative Ames, we still have to test it in vivo. And if that in vivo data comes out to be positive, we don't know how to use that currently because there's no acceptable methodology. Only if everything is consistently negative can we use that. But that's not going to be the case for 50% or more of the NDSRIs, and then we're stuck with all the others.
Yes, well, if you're writing a guideline, that's where scientific judgment comes in. That's where the application of that comes in. And if there's something about the human metabolic activation that's unique to humans, I think we should pay attention to that.
Thank you. So I realize that we're towards past time. Now, I see that we have one question from our online, and so I'll turn it over to Dr. Keck.
Thank you, Dr. Dorsam. Yes, so I'm bringing forth a question from online, and this was directed at Dr. Heflich, but perhaps Dr. King might be better suited to answer this one, and that is, if an Ames test outcome is positive, is there a chance that an improved and extended Ames test protocol for NDSRIs can be used as a basis for a compound-specific acceptable intake?
Thank you for that question. So the Ames test is a hazard identification test, and so it would indicate whether something is mutagenic or not. And so that alone is not that alone will not lead to a calculation of acceptable intake. So as described in the ICHM7 guidance, so the acceptable intake is calculated from the TD50 value. Dr. Kruhlak very nicely explained it in her presentation. And so the Ames test alone, whether it's enhanced or not, that outcome itself is not going to derive an acceptable intake.
And I think it's worth adding here that you can theoretically get a mutagenic potency from a modified Ames protocol if you start looking at dose responses. But there's the step between the in vitro bacterial mutagenic potency and a putative human carcinogenic potency is substantial. Whereas the step between, if we're to move to say a TGR, is then an in vivo mammalian system, and the potency should be a lot more comparable.
Yes, I agree with David totally on that. I wouldn't do any quantitative risk assessment based on Ames data.
Thank you. So I realize that Sorry, Dr. Teasdale.
Yeah, no, it's just a final thing I was just thinking about reflecting on as regards this that we haven't really touched on in this discussion, but was involved quite a bit in the HESI FDA discussion, was the ultimate move away from the transgenic study to actually moving towards duplex sequencing. And I suppose, that is something that maybe offers a lot more opportunities, both in terms of the capacity to do the testing, but also as well with the actual sequencing from the sequencing itself to be able to look at the specific sites of mutation and ultimately define potency on the results of that as well. So, I mean, it's not so much a question, more a reflection of what I think is a very promising way forward if we can all agree, particularly in terms of expediting some kind of agreement around what a protocol should look like for that test.
Okay, point well taken. And so one of the take homes on the duplex sequencing is you're saying a need for a standard protocol?
Yeah, there was a lot of discussion of that at the FDA HESI meeting, and we can't really rely, for instance, on this being developed through what we normally do, which is the OECD. I think this is probably going to need industry and industry and regulators to really work together in harmony to try to move towards a protocol and define it, within our ourselves as regards that. I don't know if others want to comment on that, but I thought that was a really positive potential way forward as regards a lot of this, particularly given the capacity or lack of it with transgenic studies.
Yeah, I think there are a lot of issues here that people sort of gloss over when they refer to duplex sequencing. I mean, you're talking about a commercial product that's essentially a black box in a lot of its operations. And I think from a regulatory standpoint, we need to know a lot more about what is going on to make to generate a mutation frequency. We've done a lot of work with error-corrected NGS. I'd rather call the generic approach to direct analysis of rare sequence changes than the duplex sequencing, which is a trademark kind of procedure. And there are a lot of things about it in vitro studies that we've uncovered that are sort of troubling. And I wouldn't want to jump on the duplex sequencing bandwagon just quite yet before we understand a lot more about what's going on in that black box.
Interesting.
Okay, well, thank you very much. I want to thank our speakers as well as the participants for all of their very insightful questions and for this very helpful discussion. Thanks also to the CRCG for helping provide a forum for this discussion.